started. Excuse me. Almost done with the exam. I've got a faculty meeting at 11. So unless you hear otherwise, shortly after 11, when I'm on my way over to the meeting, I will drop those off in Natalie's office. You can pick them up. I already sent out an answer key for that, so if you've looked at that, you have some sense of what the expectations were. Today, we're going to do part of the review for the final. Remember, the final consists of two parts. There are 10 multiple choice questions, and yesterday I sent out a list of 10 areas that those multiple choice questions cover. This is comprehensive, so this is stuff from the beginning of the semester, but that's just 20 points worth of the final, and we're going to go through some of those concepts today with some clicker questions. Tomorrow, there will be a review for the lab portion of the final, and the lab portion of the final is 45 points, and so your total final is 65 points. And the lab portion of the final will cover the P-Glow, and it will cover the Grizzly Farm labs. Were you going to pass out the study guide, or just because it's on the website, we'll leave it there?
It's on the website.
Okay, I'll show you before we leave today, there are some resources for studying for the lab exam on the website, and then tomorrow we will go through some sample questions, and there would be time for you to ask questions as well. The final exam is a 2-hour exam period. You're not going to need 2 hours for it, but if you're the kind of person who likes to be very thorough and take all the time in the world, we'll have plenty of time for the final exam. Okay? Okay, I am not going to post the answers or these questions on the website, so please pay attention today. And I would like you to kind of think about the concepts that we are trying to distill in these particular 10 areas. And so pay attention when we go through the choices of the answer and think about kind of, and try to remember back when we covered this material and how this relates to other stuff. So this is kind of the comprehensive portion of the final exam. Again, only 20 points, so that's not a big hit on your grade, but we do think that there's some things that you should carry over. Okay, so if you would go ahead, if you haven't responded to this first question, do that. Again, when responses get above about 35, I'm just going to go ahead and shut it off so we're not waiting for everyone all day. I'm going to grab a clicker here. Okay, good. Majority of the class realizes that the organelle that takes up much of the volume of the plant cell is the vacuole. What's that vacuole filled with? Primarily water, and so that exerts pressure from the inside of the cell out. What's that going to do to the cytoplasm? Got this big kind of balloon-like structure in the middle of the cell. What's that going to do to the cytoplasm? If it's the vacuole's inflated. It's going to push it out toward the edges, right? So there's really going to be a very thin little strip of cytoplasm. Is there cytoplasm inside the vacuole? No? Okay. So really, for a big, large plant cell, there's relatively little cytoplasm because of the presence of that large vacuole. Okay, good again. What's the issue that limits plant and animal and bacterial cell size? It's surface area to volume ratio, right? And what's the surface area? The surface area is really how much membrane you have. And why is that important? Because you've got to move things across the membrane. You've got to move nutrients from the environment into the cell. You've got to get rid of waste. What are we talking about over here? We're talking about the cell's metabolic needs. Well, that's the cytoplasm. That's the volume, okay? So we've got to have a sufficient surface area to volume ratio. What happens to that ratio as cells get larger? Surface area to volume ratio decreases. Why? Because the surface area is increasing by the square of the size increase, and the volume is increasing by the cube of the size increase. So as you get bigger, that ratio becomes less and less favorable, less and less membrane to bring things in to support the metabolism in the volume or the cytoplasm. Okay? This one down here, yes, plants are surrounded by rigid cell walls, but cell walls can expand, so that doesn't limit cell size. Okay, we're going to roll. Okay. Can plants magically say, this doesn't apply to me, I don't have to worry about a favorable surface area to volume ratio? No. All cells are bound by this particular restriction. The reason plant cells can be a little bit larger is that they really have a reduced cytoplasm, if you think about it. That vacuole is taking up much of the room inside that volume. And so the surface area to volume ratio is still favorable because even though there's not as much surface area relative to cell size, there's not as much volume relative to cell size either. All right, 100%. What could I have added as another site of ATP synthesis that I've chosen for some eukaryotic cells? Chloroplasts. Remember, chloroplasts also produce ATP? But that wasn't one of your choices. This better be 100%. Oh! Chloroplasts, right? Can produce ATP, produce oxygen, and reduce 10-ADP. ATP in the light, chloroplast. ATP in the dark, mitochondria. Okay, remember chloroplasts use photophosphorylation, light-dependent. Mitochondria don't have that requirement. So make sure you read your question carefully. It says those fractions are to contain respectively. So what's the first one? What's the second one? Okay? Class is still correct, but there's some misconception down here. Ribosomes are in prokaryotic cells. Mitochondria? No? 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 No. They don't have those membrane-bound organelles. Okay, what knocks out A and E? Because bacteria is any kind of organism. That knocks out A and E, right? Nearly any eukaryotic organism? No. An animal, but not a plant. This implies that plants don't have mitochondria. Do plants have mitochondria? Yes, they do. So it could be any multicellular organism, such as a plant or an animal. I suppose, okay, I guess you could make an argument for C. What do you think, Dr. Morden? You make an argument for C? Nearly any, I suppose you could say. No.
What would the exception be?
Yeah. No. An exception that didn't have.
You don't have to be multicellular to have it start. So
Right.
It seems to me C would be the more reasonable answer.
Let me think about that one.
That's who mine would. Okay, in order to be a cell, you need all these except. Are prokaryote cells? Do they have mitochondria? Nope, this is one of those membrane-bound organelles. To be a cell, you definitely have to have a membrane, you have to have ribosomes, you have to have DNA, although it doesn't have to be enclosed in a nuclear membrane, and you have to have cytoplasm. Okay, remember the fluid mosaic model. You've got your phospholipids making up your bilayer. Hydrophilic, hydrophobic. And so this one's wrong. They don't have hydrophilic tails in the interior of the membrane. They have hydrophobic tails in this hydrophobic environment. Can lipids move laterally from side to side? Yes, they can. In fact, they do that very readily. Okay? Do they frequently go from this side to this side? Flip-flop. No. I mean, it's possible they can do it, but it's really, really rare. Why? Because you'd have to rotate this hydrophilic head through this hydrophobic core of the membrane. Okay? Do these phospholipids all of a sudden just leave and kind of go out in here into the solution? No, they're hydrophobic. They don't want to be in that surrounding solution. Okay, so the correct answer is A. They can move laterally, and they do so very readily. Correct answer is D. The reason they're able to move from side to side is that there are only weak hydrophobic interactions in the interior of this membrane. So there's not things going on in here that keep the lipids from moving laterally from side to side. Is the interior of the membrane filled with water? No, water's excluded. That's a hydrophobic environment. Do lipids and proteins repulse each other? No, repulsion indicates opposite charge or like charges, and lipids aren't charged. Are the hydrophilic portions of the lipids in the interior of the membrane? Hydrophilic, no, hydrophobic. And then there's no cellulose in the cell membrane. Okay. Can membrane proteins move within that membrane too? Potentially, unless they're anchored by the cytoskeleton. So that really is a true statement that they can move. It's not true that phospholipids can't move within this bilayer. Okay, membrane is most permeable to small things and things that are hydrophobic. Remember, the challenge is to get across this hydrophobic barrier, like dissolving like. Monosaccharides are getting a little bit big. Even small ions can't move through this membrane effectively because of the hydrophobic core. And large things move slower than small things. Boom. Okay, correct answer is D. These are all forms of passive transport. Passive transport involves movement of something down its concentration gradient. Facilitated diffusion moves from high concentration to low concentration. Diffusion moves from high concentration to low concentration. Osmosis is a special case of movement of water from high concentration to low concentration. What's the difference between diffusion and facilitated diffusion? Requires a protein carrier because the membrane is not permeable to the substance normally. But it's still a form of passive transport. Okay, this is a question you have to break down a little bit and kind of look at what's being given here. So where is stuff moving? It is moving from the glucose-rich food, which is outside, into the glucose-poor cytoplasm. So what direction are things moving? High glucose concentration to low glucose concentration. Okay, so is that active transport? No, we've eliminated active transport. Okay. It's coming in, and so would that be exocytosis? Exocytosis moves things out, so we can eliminate that one. Is it phagocytosis? Remember, phagocytosis is a bulk transport where we move big things in by pinching in the membrane. This is not bulk transport. So is this simple diffusion or facilitated diffusion? We're down to two. What does this tell you here? Glucose diffuses slowly through artificial phospholipid bilayers. So what does that tell you about normal permeability of just a regular old phospholipid bilayer to glucose? Not very permeable, right, if things go through really slowly? So we got an impermeable membrane and something moving from a high concentration outside to a low concentration inside. That's going to be facilitated diffusion then. Why do these intestinal cells move things rapidly where the artificial phospholipid bilayer doesn't? Because these cells have that carrier protein that can move that glucose across the membrane. So make sure that when you read questions like this that have a lot of information, you kind of break it down and indicate, okay, what am I being told by this information? What are the relevant points? Correct answer is B. ATP is so important to metabolism because it provides energy coupling between exergonic and endergonic reactions. Its terminal phosphate is a weak covalent bond. It's a very unstable covalent bond. And because of that, that phosphate bond is broken off easily. Okay, so this is kind of the energy intermediate that allows exergonic reactions to drive endergonic reactions. This is the correct answer. Can some of those phosphates that are be used to make more ATP? Can it be stuck back onto ADP? Yes. Is that the only thing they're used for? No. In fact, the major reason that this is an important molecule in metabolism is because that terminal phosphate is so unstable that it's easily broken off. And then when that phosphate is released, it can be used to phosphorylate other molecules and change their shape. That's how the active transport pumps work, remember. That's how contractile proteins work. Okay? Can heat be used to do meaningful cellular work? No. Low temperature heat is considered to be a waste form of energy. So what do we have to do? We have to generate it out to the environment. When we dump that heat out into the environment, it increases the entropy of the surroundings. And that's why cells can live, is that they maintain their state of low entropy by increasing the entropy of their surrounding environment. And much of that is heat energy that they get rid of. Glucose loses hydrogens and becomes oxidized. Oxygen picks up those hydrogens and becomes reduced. This is an oxidation reduction reaction. It's the only place where it's directly involved. Remember, at the very end of that electron transport chain is where the oxygen is functioning. Will the citric acid cycle stop in the absence of oxygen? Yes, it will. But why? Because the electron transport isn't working and isn't recycling those coenzymes. And it's the unavailability of those coenzymes that stops the citric acid cycle. But if you look at all these sequences, no place oxygen directly involved except the electron transport chain. Okay? Terminal electron receptor in the electron transport chain. Add some electrons and some protons to it, and you form water. Enzymes do indeed increase the rate of the reaction, but they do not alter the delta G. They don't change the free energy of that particular reaction. Okay. Remember, this was the whole point of your lab exercise and your lab project, to see what kind of physical parameters affected the rate of enzymatic reaction. The increased rate by lowering the energy of activation barrier. Enzymes don't provide energy to the reaction. They lower the energy of activation barrier. It's an important distinction. So in your Guaiacol tubes, did you get a factor reaction rate when you included enzyme in it? Obviously, yes. You increase the rate of the reaction. DNA, RNA, protein. Previous slide, DNA transcribed into RNA, RNA translated into protein. That is what the central dogma says. Genotype is your DNA. Phenotype is really determined by your protein. So the central dogma again. Okay. Again, remember, total's got to be 100%. So if you're 30% thymine, you're 30% adenine, A equals T. That's your 60%. So there's 40% divided between the other two nucleotides. 20% of G and C. And if you don't remember your math rules and things like that, we know A equals to T, G equals to C. Put in some numbers there. Okay, if you say just arbitrarily it's 30% adenine, that's 30% thymine, therefore 20% that, 20% that, 50% equals 50%. Okay, again, this mathematically does not work out. If this is 30, and that's got to be 30, that's 60%. 20, 20, that's 40%. 60 doesn't equal 40. Okay, so there's some areas you seem to be doing well on, other areas where there seem to be some misconceptions on. So please look over that sheet that I gave you. And those are the same 10 concepts that we've been kind of beating to death today. On the cell biology website, there is a thing down at the bottom which says lab exam 2 study materials. I have the PGLO questions that you turned in with answers for those, and so if you need to refresh your memory about the PGLO, I think this is Dr. Morton's slides that he gave over the PGLO and Grizz Farm Labs introduction. This is kind of what we're going to be working on tomorrow, although we may modify it a little bit. And then there's a study guide. Oh, did you give them the scramble already?
Yes, well, except for today's group.
Okay. So there's an answer key to that scramble. And then there's a lab final study guide Dr. Morton just put together.
Please bring your lab manual to class tomorrow. Bring your lab manual to class tomorrow.
So if you can look over some of these things, particularly the study guide, I think that the review tomorrow will be more meaningful for you. Okay? So just a little bit after 11 today, you should be able to pick up your exams.